Hello, Colby Deal with the Protein Synthesis Storybook project. Getting right into the subject. For page 1, I have the DNA strands start to unwind from the enzymes that have been put on it. Polymerase, the DNA enzyme, helps unwind and copy out the nucleotides in the DNA, which helps create messenger RNA. Here I have a little picture of the DNA and the RNA being created off of the polymerase. Next, the message inscribed in the RNA codes for the specific amino acids that will be put into a protein later on. Here I have a picture of the RNA with certain codons, which are in pairs of 3 that code for amino acids. The message in the RNA strand travels out of the nucleus once it has been copied, and from here it goes to find a ribosome. I have a picture of the cell with a piece of DNA and RNA leaving it, going and finding a ribosome. Next, once the RNA finally finds a ribosome, it then becomes bonded with it, and the ribosome essentially reads the RNA. Using the help of transport RNA, it gathers amino acids which help form the protein. You have a picture of the ribosome gathering the amino acids with the help of transport RNA. Next, all the amino acids have been added together. This creates a polypeptide, which basically is a chain of amino acids. From here, it'll keep going until a specific stop codon is read. Here I have a picture of the ribosome and a chain of amino acids coming off of it. From here, once that stop codon is read, then the transport RNA will stop transporting amino acids, and the messenger RNA would break off from the ribosome, and so would the amino acid chain. Here I have a picture of both of those separating from the ribosome. These polypeptides, depending on the amino acids in them, can form very specific shapes. These become the proteins and can thus perform their functions based off of their structure. Here I have a picture of the amino acids slowly and naturally trying to gain their shape. Next, the amino acid determines the shape of the protein, which can make it an enzyme, which is the more common use, or it can simply be used for other purposes around the cell. Here I have a picture of the protein slowly beginning to wind and getting into its proper shape. To describe a few uses of these enzymes that we have, commonly enzymes are used to break down certain chemicals or materials which the cell can then use for its own benefit. Picture of an enzyme, it breaking down specific chemicals. All of these things are only possible with the help of RNA and DNA sequencing, which is where the mRNA strand and useless parts are cut off from it. Meanwhile, the useful parts are kept and thus read by the ribosome. You have a picture of those parts being separated from the good parts. And that concludes my protein synthesis storybook.